Hello and welcome to the ChemCast. I'm George Strompalompalus. With me is Hermit the Frog.
And today we are here to talk about Adriamycin.
Adriamycin is a drug used in chemotherapy, and we'll discuss the history, how it's made, and uses of this drug.
Yeah, so to start, the history. In the 1950s, there was an Italian research company called Pharmitalia Research Laboratories, and it was looking for an anti-cancer compound, and they were looking specifically at soil-based compounds. The sample was taken from the Castel di Monta, and it was a 13th century castle, and after some experiments, they managed to isolate a compound called Streptomyces pucetus, which was it was very good against mouse tumors. So the drug, it was successful at treating acute leukemia and lipoma, but the problem was that it could cause cardiac toxicity, which was really bad because it killed you.
It was fatal.
Fatal, yeah, exactly. So the structure was tempered with a bit, and a new strain was created called Adriamycin, which was later changed to Daunorubicin. So do you want to tell us a little bit about how it's made?
Well, they take Daunorubicin, which was the original compound that they found, and they hydroxylate it. So Daunorubicin is found naturally in the soil, and the formula is C27H29NO11. Due to its extremely short half-life, which is less than 24 hours, the drug must be part of a liposome and then extracted when about to be used.
Yeah, and it's kind of weird, but the geometric equation for or name for it is huge.
It's very huge. Like
Yeah.
our old science teacher, Mr. Wallman, when he was telling about those university ones, it looks like one of those.
Yeah. So let's talk about a little bit how it works. So they're not entirely sure about how it works, but they know generally that it attaches itself to DNA and it stops it from biosynthesis, which is when the DNA replicates itself. So this makes it so that the cancer can't spread, and the way it does this is by interacting between the base pairs of the DNA.
Yeah. The what's it called? Those base pairs.
The A and the T, the adenine and thymine, and the guanine and cytosine.
Yeah. So it's used in leukemias and Hodgkin's lymphomas, as well as cancer of the bladder, breast, stomach, lungs, ovaries, so on. It's normally used in conjunction with other drugs such as cytoxin, which kills the cell quickly while the adriamycin stops the cell from reproducing. It's basically a countermeasure just to make sure everything is out of the system.
Exactly. So some of the risks associated with this drug is, first of all, heart damage, because like the original drug, it's still not perfect at keeping the heart from being damaged, but it's a lot better, and with short amounts of it, small amounts of it, it shouldn't affect you much. It can be a cause of typhus, which is a life-threatening bowel infection. It causes nausea, vomiting, which is seen a lot with chemotherapy drugs. It decreases white blood cells, again, something pretty common with these kinds of drugs. And in some cases, it can even cause PPE, which is the rupturing of the skin on the hands and the feet, so your skin starts to actually peel off.
Mm-hmm.
Some other things?
Well, if one's had hepatitis B in the past, it could resurface. So hepatitis B is a risk. You probably won't be taking this drug if you've had hepatitis B in the past. The doctor will inform you of this. And the urine can be discolored, turning a very bright red, which is because of the drug's color.
Luckily, though, it's not blood. Important. Some people freak out about it. Go to the doctor. Don't worry. It's just the drug. So, what are some solutions? Because we have all these bad side effects. I mean, obviously, it's saving people's lives, but what kind of things can we do to stop these negative side effects?
Well, scientists aren't really sure with this drug particularly, but we're always looking for something that can help with this drug, that can reduce these negative side effects. For instance, keep your heart alive, I guess. Like, get rid of those negative side effects. There are other forms of treatment also, like surgery, taking out the tumor, radiology, those kind of things.
Yeah, and it's kind of interesting too. They're looking into things like cavitation bubbles to destroy them, or proteins that will target them. So, yeah. So that's why we do things like the Terry Fox Run and stuff like that, funding for cancer research, because it is expensive and it is
takes
important.
a lot of money.
Yeah. So that wraps us up today at the ChemCast, but we hope to see you next week.
See you then.